So you must have seen this kind of images in research articles. This is in vivo imaging of live animals like mice, rat. So this you can achieve by using certain dyes that you can inject via intravenous route, and then you can see the tissue localization of particular whatever you are injected or whatever you want to see. So in which part of tissues or organs it is deposited. So this is biodistribution and all these kind of things you can see using this. So for this you need to have the dyes which are in near infrared region. So some of the examples of dyes which are in near infrared regions are cyanin 3, cyanin 5 and indocyanin green. So ICG dye is most commonly used dye. Apart from that, why you need infrared light? Because that the tissue is going to, otherwise if you used other dyes, so their color will be same as that of blood, or you will not be able to see it. So this particular dye selection is very essential, and these cyanin dyes are useful if you want to tag the some nanoparticles, so that you can do. So this is one of the example indocyanin green, that you need to either you can inject this via intraperitoneal route, but you need to see for the confirmation in various research articles. So if you have an idea what other dyes you can use, then please mention in the comment box. So your aim is to visualize this in the internal organs particular, so how that particular thing is going in the circulation, and then in which organs it is getting deposited, for how long. So time intervals you can do the imaging, for example 4 hour, 8 hour, 12 hour, and then you can see. So cyanin 3 and cyanin 5 channels dyes you can use if you want to target some nanoparticles to the brain or any other organ. So you can conjugate the nanoparticles with these dyes, and then you can inject via intravenous route, and then you can see how much is the half-life excretion. Then particular, for suppose some dyes or for example nanoparticle are staying for long time in particular organ, then you can say that these nanoparticles are also responsible for causing toxicity, because they are not getting easily excreted above if they have half-life above certain level. So for that you need to do this biodistribution study. Apart from the whole body imaging, you can also isolate these organs after sacrificing the animals, and then you can see, because if you see whole body imaging, then the organs are over another organ, so you will not be able to tell exact location of the organs. But if you take these organs, isolate these particular organs, and then ex vivo bioimaging of these organs you can do. So this is also a good option. And then also these organs you can take the homogenate, or you can do various treatments with that organs also. So for example metal concentration you can determine, or various things you can do. So this is important for, particularly in vivo imaging is important for determining the pharmacokinetics and biodistribution of nanoparticles, and not only nanoparticles, whatever you want to see, but the condition is you need to tag it with the dyes, or it should be comparable. So you need to also have the control group where you are injecting dye alone, so that will give you an idea so how much the half life of particular dye, and it is because of dye or it is because of your nanoparticles or a drug that is responsible for the for that kinetics or toxicokinetics or biodistribution of that particular thing. So there are various kind of studies you can perform. So this is all about in vivo whole body imaging. So what do you understand by that? This is because uh, this is updated, or there are lot and lots of studies are being done. So this is one of the approach in apart from routine tests like determining the concentration in plasma or serum or in uh, directly in tissues. So before going to perform that, you can just see for the whole body or whole imaging without killing the animals, so that after the washout period you can also use these animals for any experimentation. So this is one of the approach you can say, where you reduce the number of animals used in the study, so the remaining animals you can use for other studies. So how this in vivo imaging can be done? So this is the instrument of for performing in vivo imaging, that is Perkin-Elmer is one of the distributor or company that supplies these instruments. So there is nothing much is there in the instrument, you just need to keep it in the, but you need to choose the background or where you are keeping the animal, like white sheet or particular sheets will be provided, and other rest of the job will be performed by the software, where what you need to measure and how the settings are, that you need to just know, and the operator for the initial days, that operator will guide you on the initial days when you perform. But before directly performing this in the like rat and mice, you need to shave the area where you want to image that particular, because these furs are there, so many furs are there on the rat, so that will actually give background color. So they also, suppose blood component is also one of the thing which gives red color, so for that you need to choose particular dyes. So hair removal will be done either by shaving, or you can use any uh, epilatories, or by using depilation method. So that way whatever you want to do, you can perform as per your choice. Next is result showing. One data you will get in the form of the left side black figure that is shown, like it will just give you an idea where this signal is coming. But while doing quantification, though, so you need to represent this data in the quantitative form by using total photon flux. So this you can see in that software settings, and then you can particularly quantify the data, how much is the photon flux for each group and each animal. So this is how you can count it in, or other thing is in terms of fluorescence intensity, this is how also you can show your data. So these are the two ways how you can show your quantitative data. So if you have an more idea about how to show this in vivo imaging, biodistribution, or any pharmacokinetics, dynamics, or whatever, what you can get, what other parameters you can evaluate or get with the help of in vivo imaging, that you can share with us, and also how you are going to represent this data. So what is the validity of this data, is there any flaw in the, means what is the assurance that you are going to record the data the exact manner what you wanted, and all that you can share. So this is also really very good